Hi, everyone. Welcome to this plenary session. And I'm really glad to have the opportunity to introduce our invited speaker. I'm Fei Fang from Carnegie Mellon University. And today we have Professor Regina Basile to talk about her research. Regina is a professor in the Department of Electrical Engineering and Computer Science and a member of the Computer Science and Artificial Intelligence Lab at MIT. So her research interests are in natural language processing, and currently she is focusing on bringing the power of machine learning to oncology. And in collaboration with physicians and her students, she is devising deep learning models that utilize imaging, free text, and structured data to identify trends that affect early diagnosis, treatment, and disease prevention. And she is playing a leading role in creating new models that advance the capacity of computers to harness the power of human language data. She has received a lot of awards, including the MacArthur Fellowship, the NSF Career Award, and the MIT Technology Review TR Under 35 Award, and Microsoft Faculty Fellowship, and several Best Paper Awards in top natural language processing conferences. And in 2017, she received the MacArthur Fellowship, which is and an ACL Fellowship and a AAAI Fellowship. Most recently, in 2020, she is the first recipient of the AAAI Squirrel AI Award for AI for the Benefit of Humanity. And she has prior to her career in MIT, she received her master and bachelor's degree from Ben-Gurion University of the Negev, and she received her PhD in computer science from Columbia University and spent a year as a postdoc at Cornell. So I'm really excited here as first as the one who introduces Regina, and also as a co-chair of the AI for Social Impact track for this year's AAAI. And I'm really excited to see how Regina's work impacted the society, and really looking forward to her talk. So with that, let's welcome Regina. And for the audience, you can ask questions through the Rocket chat, and we will ask questions after her talk.
Hello. First of all, Faye, thank you very much for the introduction. I really appreciate it. And thank you for all of those who decided to take the time this evening or whatever time it is in your time zone and spend it listening to a talk. So as Faye said earlier, my first area where I started my work in AI is natural language processing. And maybe 5 or 6 years ago, I branched out into two areas. One is drug discovery. Another one is cancer diagnostics. And it was really astonishing to see how much these areas grew in this relatively short time. And I was really blessed to be able to contribute to these changes. So I decided to talk about these two areas just to give you an idea about the landscape of research in this area, but more importantly, highlight the questions which are still unanswered, where we need to have new algorithm, and this algorithm can really make a great impact on the applications. And in the final few minutes of the talk, I would answer questions that people typically ask me is, how is it to transition to new fields? And why did I do the transition? But first, let's start with the technical content of the talk. And the first area I will talk about is drug discovery. So I'm pretty sure that every member of the audience, no matter what is your background, knows that developing of drugs is extremely slow and expensive. It's really mind-boggling that, to put a drug on the market costs several $1 billion. And there are lots and lots of diseases for which we don't have any good cures. So this is really an area where we want to see impact. Now, today, drug discovery is primarily experimentally driven. There is a lot of efforts where you take the molecule and you empirically investigate its properties, and afterwards, you continue your analysis. So the problem here is that the space of molecules is actually humongous, and you need to go through this humongous space to find just one good candidate. And many times it's almost random search. There are multiple publications that try to quantify what is the space of druggable molecules. So the numbers that I recently seen is 10 in the 60s. Now, even though you can do the screening, high throughput screening with robots and make it fast, we're just talking about millions of molecules. So there is no way you can empirically investigate this humongous space. And that's exactly where we can use machine learning to enable us to explore in silico virtually all this huge space and then just test a small subset of these molecules. So one interesting thing to note that I entered this field in 2015. The interesting part is not that I entered it, but in 2015, there was almost no work on modeling chemistry with machine learning. And here I just collected some statistics from ICLE and NeurIPS. There are many other conferences, of course, but you can see 2015, there are no papers. And every year we see increase in papers. So it's still, of course, not as big as NLP or computer vision, but at least the gradient is, goes in the right direction. But the question that you may be asking yourself is, okay, fine. We are writing papers. Do we really make an impact? Are these techniques that we've developed in the last few years can really do drug discovery? And I'm happy to share with you that a number of collaborators, including me, use machine learning to develop this new antibiotic, which is called calycine. It has its own Wikipedia page, and you can read about it. So this antibiotic is highly effective against a variety of pathogens, for some of them there is no good therapeutics today. And we had a paper in Top Biology journal Cell, which described the discovery and the properties of these antibiotics. Now, I would say that we were, again, beginner's luck. We were actually very lucky to look at the area of antibiotics. So why a machine learning can make here a difference? Because for those of you who don't know, bacteria evolve very fast, and they evolve, the resistance continues to grow, and our development of the drugs is really, really slow. And what happened, if you look here in the this depiction on the slide, you would see that majority of antibiotics were actually developed decades ago. And in the last century, we actually haven't seen a lot of new advancement and a lot of drugs. And one of the reasons that economically it's not worthy for pharmaceutical companies to work on antibiotics for various monetary reasons. So this space is currently a space of void. So the funny part about this calycine, that actually discovery here really happened on the budget. Of course, we don't have $1 billion to put a drug. We don't even have $1 million. So what we did have was like a very small amount of money. One of the postdoc who really led the work on this project, his name is Jonathan Stokes, he scanned in the lab 2,500 molecules against E. coli, which is, one of the strains. What you can see here on the left, it's how the screening looks like. It's actually, if it is kind of opaque, it means that the drug didn't work. And if it is transparent, it means it did work. So at any rate, what you get after the screening is for each molecule that you try from this 2,500, you can measure, did it actually help to did it decrease the growth of the pathogen or not? And then you can just take the neural nets, like we'll talk about architectures later, but you can just look at the molecule and the number and train your model. So at any rate, the assumption that we are going to be making is kind of very simplistic assumptions. We would say, okay, so Jonathan Stokes went and screened these molecules, but we don't care really. What we do have is a 2D representations of the molecules, and we have some number which reflects, let's say, inhibitory capacity of the molecule. Okay, and why is it a good way to do any learning? How we can find patterns? Because many of you who are familiar with biology say there is no biology in this depiction. So the thing that people knew in chemistry and computational chemistry for quite some time, that actually some fragments of molecule are responsible for its function. So for instance, here on the slide, you see a big class of antibiotics, and you can see that many members of this class have this particular subgroup in it. Obviously, these patterns can be much more sophisticated than just, you know, a subgraph. And the hope is that if you're provided with enough data, the model would be able to learn it. And again, the discovery in the budget, this is our discovery pipeline. So we actually screen several million molecules using our model. We empirically tested just few hundred molecules, like less than 200, and during testing, and then the animal testing was just one molecule. And we were really lucky as a beginners. First of all, we discovered that this particular molecule halicin is not only active against E. coli, but it also works against drug-resistant strains of C. difficile and A. bowmani. And you can look up in Wikipedia you know, more about these particular pathogens, but again, for many of them, we don't have cure, and they are on the list of WHO critical therapeutics list. The reason that this drug was so effective, it's actually something very much surprising to all of us who come from the AI perspective, that it actually has a distinct mechanism of action, the known antibiotic classes. That's why it could do such a great job with drug-resistant strains. Okay, so now we kind of see what it can deliver theoretically, these methods, and we can ask ourselves, what techniques are we using here? And the key technique here is actually property prediction. Again, you are given a molecule and you try to predict a property. Until now, I described you the property, which is this antibacterial property, but it can be anything else. You can measure toxicity, you can measure solubility, and many other things. And if you do have this capacity to look at the molecule in silico without actually manufacturing or doing any experiments, first of all, you can do virtual screening, as we discussed. But we also can do totally de novo design of drugs, and I will talk about it in the second part of the talk. Now, what is so new with the current technologies that people who were doing computational medicinal chemistry couldn't do before? So traditionally in computational chemistry, people really understood that substructures are responsible for properties. So the way they dealt with it, they will take a molecule and then summarize it as a vector, they're called molecular fingerprints, where every coordinate of the vector actually records some substructure. And then you are applying machine learning to learn how to combine the presence of different substructure to predict the property. And of course, it will depend on the expert. And if the expert knows about some subgroup, then they can put it into the, into this vector. And if they don't know, then we will not see it. And as you can see, sometimes experts really don't know. So instead, of course, we will do normal stuff related to representation learning. We will take our molecule and use, in this case, graph convolutions to translate it into a vector and do predictions from this vector. So when we are doing it, again, we are losing interpretability of this vector, but the hope is that we learn this translation in such a way that it is optimized for our task of interest. And again, this is kind of a dream world, a desiderata of our learning. We hope that we embedded our 2D graphs into some high dimensional space in such a way that molecules whose embeddings are close in the space have similar properties, and molecules which are far away, they have distinct properties. This is the hope. And I shall say that we developed this tool, for those of you who are interested, it's called ChemProbe, that kind of implement these ideas, and it works well across a variety of different data sets, both for bioactivity, toxicity, physical-chemical properties. And, people are using it today. It's like a workhorse in pharmaceutical industry and other places. Now you can say, wow, like so little. You just took, graph convolutions, applied to these 2D graphs, and miracle happened. So one thing to understand, that there is very serious data challenge when you are working in chemistry, because what people typically want to do is to look molecules which come from some small portion of the set and apply, generalize to other parts of the space. So you are explicitly making training and testing not random, like what we do in computer vision or in NLP when we evaluate, we typically randomly do the split. In chemistry, it's not what people do. They separate training, development, and testing according to scaffolds, which are kind of backbones of the molecules. So you really want to make sure that whatever you're trained on looks very different from what you're testing on. So we need to have techniques which can generalize well. The other big problem is that, especially when you're starting a new therapeutic program, you may have very few molecules. And we cannot assume that we have hundreds of thousands of points that we assume, in many other fields. And it's very expensive to expand it. So now I want to tell you about, what can we do? And, obviously, when you are entering such a field, you can say, okay, let me check what is out there and what I can reuse. I want to give you an example of something that was reused, adapted to chemistry and works beautifully. And I want to give you an example of something that was reused and doesn't work at all. So let's start with a positive example. So as many are aware, enforcing invariants become really important and robust learning with their algorithms like IRM and others. So here you can actually apply the same idea. You can emulate the idea of domains by just taking your training data and dividing it to domains according to scaffolds. And then you want to force your predictor to perform well on new scaffolds, even if it hasn't seen them before. The challenge here is that scaffolds are actually structured objects, and it can happen that you would have, every scaffold may be its own domain. So you need to do something smarter to deal with this combinatorial structure of scaffolds. And in this paper below, we describe how you can define domains through perturbations on the representation, which deals with this question. But the general idea that comes from, enforcing invariants applied here works well. Let me show you examples where things really didn't work well. And this is an idea that extremely popular, again, works very well in computer vision and natural language processing. This is pre-training, correct? We all use pre-training, and the way you do pre-training in NLP, you just you mask some words and try to predict them. You can do the same thing in molecules. You can just mask either individual atoms, or you can mask some substructure, try to predict it back. Lots of people tried it. And of course, there is a huge space of unannotated molecules. Nobody made pre-training work for molecular tasks so far. We didn't see consistent improvement across data sets. So this is one open question. I mean, I can speculate why, but this is a place where we can really make a big difference to utilize all the data to do better on our prediction tasks. But of course, there are lots of other ideas that we can directly incorporate from, from other areas of machine learning and just say, let's reuse and recycle them. But let's just go back to the original question. Let's just think about our formulation. And in this case, how did I formulate the problem? I told them you have the 2D, a number, and let's just predict the mapping between the 2D structure and the number. But the question is, what happened to biology? Because there is a meaning to these numbers. The fact that I don't give to my models any meaning doesn't mean that it doesn't exist. So clearly, maybe if we add some biological knowledge to these models, they are going to be doing better, and they will not need so much training data. And again, if you have enough training data, if your screening data is very large, maybe you don't need biology. But if you don't have enough data, maybe this extra knowledge can help. And here again, I'm showing how much we are simplifying by looking at the, at this particular problem formulation. You can look at the normal reality, at least some reality of the biological connection. So in this case, you can see that you have actually a protein, which is a 3D object. There is a molecule, which is in yellow here, which binds and makes this protein inactive. So there is a lot of things that happen with the protein and that happen with the molecule, but we are forgetting it and just summarizing it by this number. So what I want to show you is that even if you don't model the whole thing, how this interaction happens, even if you add the information about the targets, meaning the proteins to which molecule bind, you can already improve. And the situation where we were forced to do it relates to COVID antivirals. We, like everybody else, started working on predicting COVID antivirals. And people very soon identify that there are no good single safe compounds. Safe compounds meaning either FDA approved or clinical, or preclinically tested compounds. And you can see that even the drug, which is FDA approved, remdesivir, if you look at its mortality decrease, it's actually very much not impressive. So what you want to do, you want to increase the potency. And one good way to increase potency today is to look at combination therapies. And we know that, for instance, in HIV, combination therapy is a standard of care today. So our question was how you can predict a good combination therapies for COVID antivirals. The problem is that we only had 300 screen combinations. So it's kind of hard to imagine running, a convolutional neural network on 300 combinations. The good news, however, is that people actually in biology understand a lot about viral replication cycle. They know which receptors the virus used to get into the cell, which proteases it used to replicate, how it interacts with host protein. So this information is known. So the targets were actually already identified last February. So there is a lot of understanding. There is also independently a lot of screening data that just looks at the compounds and the targets and sees which compound activate which target. So you have this information. You can relate the molecule to the target it activates. So what we would want to do is actually to expand the architecture that I just showed you. Again, very simple change. Previously, we translated directly from the chemical structure, we summarize it into a vector and got the activity. Right now, we're gonna be doing something else. We're gonna, in our representation, augment it with biological information where we are trying to predict the targets which the molecule activates. So your representation will have two parts. One part is a biological kind of encapsulates the biological target activated by this compound, and another one summarizes its chemical representation, and it would be learned jointly. I will not get into the details because they're kind of obvious what we would see here, but what it enables you to do is to go from the compound and represent it in this biochemical space where the models that would look at the combination can reason both about the structures and also about the targets, because you achieve synergy when you interact with different targets. And by kind of looking at this biochemical representation of two molecules, you can directly train on synergy. And I just want to share the results, and I want to say you would see a lot of this picture of these two guys. One of them is my student, Wengong Jin. This is jointly co-advised with Tommy Yakovlev, who is a professor at MIT, so a lot of papers, you would see their pictures. So using this approach jointly with NCATS, which is one of the NIH institutes, we were able to discover some new, very promising combination, which include remdesivir. They were interested in remdesivir, which demonstrate that remdesivir and their partnering molecules on their own are weak. However, if you pair them up with each other, what you would see is that they become very potent on a small dosage. So this is an example where, despite the fact that the co-combination data was relatively small, by incorporating biological information, you can actually achieve some very promising results. Now, I don't want to give you an illusion that just kind of naming the targets is our final goal. Far from it. It's just the first step showing the promise. And our whole view is that, molecules are 2D graphs is very naive. Every medicinal chemist would say that it's not how they're thinking about molecules. And it's also problematic because you can look at two molecules, which 2D graphs look very similar to each other, just one atom away, but they will behave very differently. And this is called in chemistry property cliff, when a tiny variation in structure results in huge variation in the property. Now, it's very hard to explain it in 2D way, but you definitely can explain when you're looking at 3D conformations of these molecules. Now, and this is another funny case because, every medicinal chemist and biologist say you have to go for 3D, and it makes all the sense, you have to. But today, the best models that we've developed are 2D models. And again, it's not because this is the ground truth, it's because we still don't know what is the best algorithm to incorporate this 3D information when the conformers and 3D structure is latent and you don't know it. So this is a very active area of research, and I hope we will see some interesting developments soon. Now I want to kind of conclude my part talking about drug discovery by looking into de novo molecular design. So you remember when I started my talk, I show you the space of 10 in the 60 molecules. And even if you have amazing virtual screening capacity, you really cannot screen them all. And typically you don't just care about one property, you want to make them potent as drugs, you want to make them safe, you want them soluble. There's a whole long list of things that you want. So maybe instead of kind of going and doing this guided search in the humongous space, what we can do is to try to just generate a molecule directly with a property that we want. And of course, there is a lot of work on generative models. So, people are generating images, they're generating sentences, so why not to generate molecules? And there are a lot of machinery which is available that we can just reuse, like CNN or RNN or whatever you like. And the first kind of generation of models that was developed in chemistry did exactly that. They say, we love these models, let's just reuse them. Now you can say, what am I going to do? I have a 2D graph. So what you can do, you can, for instance, again, go back to chemistry. There is something which is called SMILES. This is an encoding of the molecular structure in a string. So you translate molecule into a string, and then you can run sequence-based generative model. The problem with that, these SMILE strings are actually not reflective of structural similarity. So what you see here in the picture below, you have two almost identical molecules, but their SMILE strings look very different. So if you are using this method of encoding, the space that you are getting is going to be really, really messy. And, people are still trying to fix it. And now there are super SMILES and variety of other things. But maybe instead of trying kind of to reuse and recycle, we can think about doing something different, which is really custom design for molecular graphs. And there are many methods. I just want to illustrate you one. And the reason I liked it is because it sort of mirrors what human chemists are doing. So the way human chemists construct the molecule, they kind of say, okay, I want to have these several properties. I know that there are some pieces that they carry a property. So what I can do, I can just put one piece that, for instance, drive property 1. Then I want property 2. I'm going to find another piece that have property 2. And then I'm going to incrementally construct my molecule, and maybe I'm going to add some extra pieces. And through these building blocks, through these fragments, I can build a molecule as a Lego piece. That's the idea. So you need to answer, if you want to automate it, you need to answer 2 questions. First of all, what are these fragments? How do you know what are the good building blocks? Human experts know it just because they know medicinal chemistry, but as we talked before, they may or may not know. I mean, they do to the best of their capacity, but we want to do better than them, correct? So, the point here is we need to learn to identify these logger pieces which are responsible for property. And then we need to have generative model that can actually create molecule by operating in the space of these fragments rather than individual atoms. So let's start with the first question. And again, you can see the picture of these two gentlemen, Wingong and Tommy. Let's start with how we can find the fragments. And it reminds of, in natural language processing, this kind of fragments are called rationals. So we can call them rationals here. The idea is to identify a piece of molecule that drives its biological properties. And and the way you can do it, you can think about it as a discrete optimization problem where you're trying to find the smallest piece and on which, when you are running the property predictor, the property is high. Okay, so in other words, that if I just take this gray piece, run my property predictor, I will get the same score as if I ran it on the whole molecule. And you can use Monte Carlo tree search to find this kind of fragments. But you can use any rational extraction algorithms that are, there are plenty of them developed in other areas like natural language processing. Now, what you've done when you run your predictors on your data for each one of the properties, you identified a vocabulary of these pieces, the vocabulary of rationals. You have them, for instance, for property 1, you have them for property 2, and now you say if my desired molecule has both property 1 and property 2, I just need to construct from these pieces my full molecule. And as I said earlier, the key point is how to do this generation at the fragment level, not at the atom level, because you are given as a unit the fragments. And here Wingong developed this hierarchical graph representation that we use for molecules, where you have two layers. The first one describes, how atoms are connected to form the fragments, and the second one describes how fragments are formed to create molecules. And you run convolution both at the level of atoms, and then you propagate it to run graph convolution at the fragment level. And given this representation, when you want to generate, to decode, you first predict the next fragment, then you identify where it is attaches, and finally you predict the type of attachment. So in summary, what this mechanism enables you to do, it enables you to grow molecule fragment by fragment until you completed the whole construction. And this method works quite well. I intentionally didn't put the numbers because I always feel in the talk we really cannot understand the numbers. Of course, it's compared with baseline and does better than others, but you are welcome to look at the papers. And I just want to say, before I move on from this topic, about the translation part. So as I started this talk, we use this model, the first model, to develop this drug halicin. But we were very lucky to work in the last 3 years with this pharmaceutical consortium of companies that are using tools developed at MIT in drug discovery. Many of them are currently part of drug discovery, and a lot of feedback that we are getting from them helps us to understand better this landscape and improve our models. Now I want to switch to a very different topic. This is my second tale. And this one would have nothing to do with chemistry. It's going to talk about clinical AI and about trees. So, each one of us have a risk profile, correct? And this risk profile, what kind of disease we can get. And you can say, okay, whatever, I have a destiny and I'm going to get some diseases. But the truth is that for many diseases, if you know early that you're going to get this disease or you're likely to get this disease, there are mechanisms for prevention. It can be detected early and cured, while today we don't have really good mechanism to treat advanced disease in oncology. Okay? So the question is, how do we today, not we, sorry, the doctors, how today the doctors talk about risk? And I'm going to talk about breast cancer because, this is like a big one since, half of the world population can get it. And no, not half of the population. I didn't mean that half can get the breast cancer. I mean that, half of the population is screened for the disease. So the way you decide your risk is actually very simplistic. People just look at several variables like age, family history, and other things, and then you run a very simple statistical model to predict the patient risk. Now, how good these models are? AUC of this model is 0.6. And I want to remind you that if you're doing totally random guess, AUC is 0.5. If you're perfect, it's 1. So very close to 0.6. Not very satisfactory. Now you can say, the problem of this model is that it never actually looks at the image, because maybe if I look at the image when the woman made mammogram, I can say something about her risk, correct? And this idea is very old. This guy is called Wolf in like 67 was saying, you have to look at the images, prior images of women who later got cancer, and see what are the patterns. And he even says here in his citation that he used computers in 67 to do this coding. I'm not sure what he meant, but he tried. So, but we can reuse this idea now, and we can say what we can do with the image. So before computers came in, doctors, and that's what happens today. Every woman in the audience knows if she goes to get mammogram, she gets something which is called density. What actually density tells you, it quantifies the amount of fat and fibrous tissue. And it's pretty much the color. If you have a darker color, you have a fatty breast, which is considered good in terms of the cancer. And if you have very white breast, then it means that you have dense breast. It's an independent predictor of cancer, and it also makes it easier to miss the cancer because cancer is white and the image is white. Okay, so what we are saying here, it doesn't matter if you, don't care about the explanation, but the point is we're taking this image, which has so many pixels, and we summarize it into 4 numbers. And then we feed them in the model, and surprise, surprise, yeah, it helps tiny bit. We now move from 60 to 63, but we're still very far from where we want to be, correct? So the question is, can we do better? And what does it mean to do better? Is to say this is like a really, really simple modeling task in contrast to chemistry. You can say, I'm going to look at the image. And my prediction task is to say whether this woman within the next 5 years is going to get breast cancer. And how can I get the training data? I can look all the women from 2016 and prior to that, for whom I know the outcomes, and some of them developed cancer, some of them didn't, and I'm just going to train my model. So we just recently got a paper in Science Translational Medicine that described all this modeling effect in detail, but I just want to mention that you really, in order to make this model robust and useful in practice, it is not enough just to take kind of standard computer vision architecture and apply. You need to model risk progression over time, make sure that I'm not sure what happened to this word in the middle. You need to enforce invariance across devices, and you need to intelligently combine multiple views. Again, not sure what happened. But I wanted to give an opportunity to a person who actually developed the model and deployed them, to my student Daniela, to tell us about this model.
Was designed to predict risk at multiple time points, leverage optional risk factors if they're available, and to produce predictions that are consistent across mammography machines. We trained the model on a large data set from Massachusetts General Hospital and tested it in held-up populations from Mass General, Karolinska in Sweden, and Chang'e Memorial in Taiwan. And across all 3 data sets, we found that the model significantly outperformed all prior methods. We also found that MURAC performed consistently and similarly well across different races, ages, breast density categories, and different cancer subtypes. This kind of detailed international validation is critical to make sure the model is robust and doesn't create or perpetuate any disparities. This is especially important given that some patient populations, like African American women, already suffer from increased breast cancer mortality.
African American women continue to present with breast cancer at younger ages and often at later stages. This, coupled with the higher incidence of triple-negative breast cancer in this group, has resulted in increased breast cancer mortality for African American women. The need to address this health disparity is apparent. This study demonstrates the development of a risk model whose prediction has notable accuracy across race. The opportunity for its use clinically is high.
So you can, you've seen here the developer, the person who developed it, Daniela, who developed most of our models and deployed them. And as I told you earlier, I don't like to put numbers in the talks, but here I do want to put at least some of the numbers. And this is really a serious concern when you're doing the clinical AI. We are dealing with a significant distributional shift from training. So if you, and that's what FDA is also obviously considering, that you are training your model on one type of data, and then you apply it inevitably on some other patients. So it's really important for us to see that across different types of population, this model work. So Adam already showed in the talk that we apply this model in 2 other countries and with quite different profile of women. Another interesting number that I want to show you is how the performance is stratified by race. So people are always talking about AI and AI that introduce bias. You can see that in this case, when you look at white women, African-American women, and Asian women, the performance is very similar, it's consistent. However, the model in blue, which is called Tyrer-Cuzick, it's a model that is currently used in clinical practice in the United States. You can see it's not a machine learning model, it's kind of standard statistical model like the one I showed you. You can really see a serious bias that this model introduces for both African-American women and for Asian women. It's close to random. So this is really our opportunity to bring these tools and to help the population that are currently at disadvantage in clinic. And this is a big concern about disparities in the AI. There is this article in the New York Times that talk about non-careful use of AI can introduce the disparities. And clearly the general population is concerned. I want to take this opportunity and to talk about our own standards of our research when we are publishing papers. So there was this paper, I think it comes from Google Health, that is called International Evaluation of an AI System for Breast Cancer Screening. This, even though it's called international, it was tested on US and UK cohorts. There is no racial breakdown provided. We don't really know, would we be comfortable to use this system that has never been tested, or maybe it was on Hispanic population or Asian population, would we be able to use it? We don't know because these numbers are not provided in the paper. So whenever we are publishing about these very sensitive topics, we really need to make sure to do our due diligence and put all the information. Another point talking about standards that I want to bring to your attention relates to reproducibility. This is another paper that published by Nature Medicine from, I think, from the same group. This paper was actually published on publicly available datasets. This is very rare because majority of the datasets that we are using, we cannot release them. This is a dataset that was released by NCI. The problem was that the results cannot be reproduced because there is no code available. There is no even information about the splits, and there is no information how the data is augmented. So there is no way you can take this paper and reproduce it. And I would tell you that after multiple correspondences with the authors, we still cannot reproduce it. I don't doubt that, they did their work well, but for the scientific community, it's really crucial for us in this new field to get to the point that we got in all the other areas of AI and machine learning where there is a clear benchmarking, and in order to publish, you need to demonstrate reproducible improvement. So we are not quite there. And finally, I was talking about the papers that I have concerns with. I want to tell you in a second about the papers that really excited me. And it actually comes from the group from another invited speaker, Sanjeev Arora, who talks about an interesting way of doing machine learning on the encrypted data. And this technology where you can take an image from an institution locally encrypted, then ship this encrypted image to another location, do the machine learning and return your prediction is really, really crucial, because one of the big impediments for doing reproducible research in this field that people cannot share the data, they cannot combine the data. So I'm really excited about this work that Sanjeev's group is doing. And finally, I want to tell you about, again, translation. So I'm happy to report that these models that we've developed actually are used in practice in Massachusetts General Hospital. And I think last week there was an article in Wired that talks about how during COVID, and this is our collaborator, Dr. Connie Lehman, is using it to decide about screening of women due to the COVID. For those of you who don't know, when the COVID came, at least to Massachusetts, the routine screening was stopped because it's considered non-essential procedure, and lots of women missed their yearly screening. So these models are used to identify women who need to be prioritized for testing, and these women are kind of put on the top of the line, and the article reports us, positive result of the algorithm. And in general, Massachusetts General Hospital uses this algorithm. Last time when I did my mammogram, I actually got the results of the algorithm. I also want to say that our goal, of course, is not only to benefit Boston area institutions. We and Adam is working extremely hard to spread this technology around the world. You can see that we're collaborating with hospitals in Latin America, Brazil and Mexico, in India, Israel, across the world, and of course in many parts of the United States, trying to validate the model and find the best uses of this technology. And I want to say that, one of the perks of being at MIT is that we have a very generous donor, Mr. Jamil, who kind of helps us to bring the advancement of AI and health to world populations. And breast cancer is just one of many examples when we are developing technology, trying to put it out for others to use. And with that, I want just to put one ad slide. In April, we have a conference, AI for Healthcare Equity. It's open to the public and free. So if you're interested in the topic, consider checking it out. Now I want to talk to you. I think, how many minutes do I have? I have maybe 5 minutes, right? So I want to tell you, how did I get to all this research? And what did I learn when I got into this field? So what you're currently seeing on the screen is my breast, clearly in mammographic form. And you can see that I was diagnosed with breast cancer in 2014. But if you look at the images that I had, the signs were already there in 2012. Nobody actually told me that I was such a high risk. And, I never looked back at my images until we actually built the model. And then it was clear that there were a lot of signs, even 2 years before the diagnosis. So at any rate, this is the pictures from this crazy year when I was diagnosed, which was in 2014. So you can see here this trio, me just months before diagnosis, and my son trying his barber skills before my chemo. And then eventually, like kind of a year after, when my hair came back, and you can see that it came curly. I didn't look like myself. It's called chemo curl. It's not a hairdresser. It just comes until it becomes straight again. And I was really lost in transition, because during this time, you are really busy. You go into chemo, you go to radiation, you have surgery, there are lots of things happening, and you are in Massachusetts General Hospital. And then at some point you're done to be treated and you go back to your life at MIT. And at that time, I had these big questions, what do I want to do? Like I was sitting there and, there were lots and lots of people, sicker people. And I was thinking, how my work, why is it relevant? That year I got some best paper award for some parsing. It's like, who cares? Who cares? I worked on undeciphered languages. Who cares? People here are really dying. And I am just sitting and doing something totally irrelevant. This was like my big lost in translation year. And I should, and for those of you who are wondering, how did I get into chemistry? I was just totally kind of in exploration mode. Somebody just asked me to join the grant in DARPA. And I thought, why not? Maybe there is something productive that can get out of it. Maybe I can have some real impact. I should say that my views were very extreme and confused, but that's how I got into it. And then, I was sitting and thinking, what do I want to do? And I was invited to give a talk in NACL HLT 2016. It's like a huge, huge honor. And at that point, I was thinking, what is it that I want to talk in the talk? I was not clear, definitely not about parsing. And in that moment, I really thought I need to talk about what the community can do to get into this field. Because before I got sick, I never worked on anything related to medical stuff. I thought it's boring. I saw that it's primarily reuse of the techniques from other fields, and I didn't see myself contributing. And then I gave this talk about, I already started working with the doctors at MGH, and I kind of shared my perspective of what I thought were important problems. And, this was really funny. So I gave this talk and people were so excited. I get all these tweets. I mean, I didn't get, people told me I didn't have Twitter then, but it was so exciting. It sounded so great. I wrote all these proposals and, I said, oh, I know what needs to be done. I know exactly what needs to be done. I even finished my proposal saying that, I'm breast cancer survivor. So I want to see it work and I know which question to ask. So I was very excited. And so this is great. And then this reality check. A year later, all the proposals that I submitted were rejected. I didn't have access to data. I was spending humongous amount of time trying to get image data. I couldn't get the image data. And, we all upset when we are getting like bad reviews. But here I just kind of felt that these reviews were exceptionally bizarre. Like, PI is a strong experience in NLP, but she doesn't have biomedical NLP. I didn't know that we have many, many different types of NLP here, So it was just really bizarre. And I remember when I was sharing my frustration with my friend Dina Katabi, she told me, who is on this panel? So I went to one of the grants, which was rejected, and they look on this panel, breast cancer panel, and there was, who is missing on the panel? It was in 2017. There was not even one AI person, even whatever we call informatics person, none. So how exactly they can accept my proposal? Now, when I was preparing to this talk, I actually thought, oh, maybe the situation changed from 2017 to 2020. Guess what? Who is still missing on the panel? Still, there is no AI on these panels. So this makes it kind of hard to get the funding. And my next plan was, I couldn't give up because I felt I have to do it. This is my mission in life. So I get 70 talks, it's like over 70 talks. I think I gave more talks. Whoever would listen, I would stand and give my presentation. Any person who is interested, I will talk to them. I try to get philanthropic money. I get more proposals. I found several like non-federal, like for instance, medical research is funded by foundations. So I wrote the foundation. I try to get publicity, and at the same time, we continue to work very hard and do tool deployment. And Adam, that you've seen previously, was a person who actually believed in me. And even when I was sort of penniless in this area of research, really try, to push this forward. And there was a group of students who, believe in me when I stopped believing in myself. And, and they will remember this picture. It's kind of a special picture because I look so happy. It was kind of the first piece of publicity that we got where people describe, what we've done. And this was a key moment of desperation. Didn't have, any funding to pay for this research. And for those of you who read this book, it's one of my favorite. It's called Americana. This is kind of some stories of desperation. That's how I felt most of the time at the beginning. Then something changed. I got MacArthur. So now I was not just, another MIT professor. I was one with with a genius or whatever. And, and things started moving. Was it MacArthur? Was it the fact that they did these things again and again and again and just refused to, to accept it? I don't know. But, definitely MacArthur helped, but hard to say. At some point, things start changing and then start moving, and we became stronger and stronger. We deployed the tools and something changed. But there were a lot of things that I learned about myself and about the world in the process. So first of all, I discovered that I'm not charming. I didn't get any money from rich people, none. Then I discovered that I really have to control what I can because I was giving all these talks and constantly engaged, and I felt I don't get money and I spend all my time. So I became like really, really crazy stingy with my time. So I very much make sure that I don't spend it on things that I don't care about. Persistence consistency pays off. So, you know, you need to try lots and lots of things, especially when you're moving to a new field, before you, you know, you see the things realize themselves, and you really don't know. When I did my NLP work, I was groomed at Columbia. I knew which papers to write, how to get the funding. It was very clear path. When you are moving, there will be a time when you don't know, and you just need to continue. Now, the other good thing is that I really don't take rejection in funding personally. In general, you know, at this moment, I feel I got so many at the beginning when I moved to this field, to clinical AI, that, you know, I'm sort of immune to them. And another important piece is really to be patient. Some data set I got after 3 years, and it's a very different cycle than we have in computer science when, you know, you wait for a few weeks, you pay mechanical Turk, and you're done in a few months. Here it can take a long time, and it takes a long time to see the things implemented in clinic. And also, you know, I learned to be humble and to understand how little sometimes I know, and you know, really understand my own limitations. You know, when I was putting the slides together, I just looked like in my notes and pictures and other things. You know, I don't know exactly, you know, how I managed to survive all these, you know, rejections. And, you know, my students were really, like Adam and others, really kind of pulled the whole thing together. But I want to tell you what was important. That the important thing for me was that over the years, because I made my story public intentionally about going through breast cancer, there were a lot of people at MIT and outside, or just normal people who I don't know who they are. They send me their stories of how they're handling their disease and how challenging it is for them, and how they suffer from the lack of technology. And, you know, and every time I read these stories, sometimes I cry when I read them, and I feel, you know, it doesn't matter how many times, you know, I get this rejection and we repeat it. You should really remember why you are in the game. And what I want to say in conclusion of my talk, that we all have different causes that move us. Some of us move for health issues, some of them about the environment. There are lots of good causes because we are all members of the human society and we all care for something. So I think that to remember what we care about and put our time, we really can see some results. And it, you know, it takes a bit of patience, but I'm sure that the internal drive and motivation can make a change. And finally, I was thinking, to whom should I thank? And there were so many people I didn't know. So I put some of them, of course, you know, all my students and my friends who pulled me through the cancer care. And my family, my son and my family in Israel. But it's really a big, big collective effort. And I am really grateful to all those that, you know, were there for me, who, you know, when I was weak, and they helped me to find strengths. So with that, I would like to finish my talk. Thank you very much.
Thank you. Yeah, thank you so much. And this is really a great talk. You've done amazing work. You have all my admiration. What a touching story of yourself. And so I'm so glad that you've done this line of research and trying and providing hope to more and more people. So we also see that there's a lot of people thank you and also giving you their admirations. And also we got questions from the Rocket Chat. So let me start by asking some of the questions listed here. So there's one person asking about the explainable AI issue. So in your work, have you experienced any case where you need to explain the decisions made by the machine learning models to the experts? And how do you handle this? Or do you need to bring the human experts in the loop in this AI for healthcare work that you have been doing?
Thank you. This is actually a great question. And I had some slides on it that I didn't include. So there are some explanation. that the doctors use and we can provide them. For instance, there is a task that many people do quite well, is like cancer detection. I give you an image, you want to identify the cancer, human doctor can identify the cancer in cycle, so you can use, you know, any attention-based model or whatever to identify this cancer and show it to the doctor. This is easy because in this case, we know exactly what kind of explanation human need to be convinced. Now, the problem is that if you're looking at the tasks like the one that I presented, which is risk, humans are not even trying to do it because they cannot distinguish it based on their eyes. The pattern is very subtle. So what exactly would it mean to give you an explanation of something that your cognitive capacity or whatever, your pattern recognition capacity doesn't enable you to capture this pattern? You know, it's the same way, you know, the dogs can smell better than us. What does it mean for them to explain what do they feel? So, and I can imagine that it's going to be a much bigger problem as we continue to be strong, to provide stronger and stronger models. How exactly machine can explain to us something that we cannot comprehend fully. And that's why, again, in some areas of clinical research, it's very clear what is the explanation. You can train to machine to provide it. But as we move forward, and there are many more these very complex subtle patterns we cannot understand, in this case, we have an issue. And that's why I think it's really important to have all these additional checks, to make sure that we can work for different populations, that we can generalize, that we work under different conditions, that we are invariant to the type of machines. These kind of things will give us much more confidence that even if we don't exactly understand what is it doing, at least we kind of have some guarantees about its performance.
Cool, thanks a lot. And another question is asking about the kind of the domain change, because it seems like in many of the cases, when taking advantage of the tools and infrastructure from one problem domain in another problem domain, then it may not work because there are implicit assumptions about the original domain, and the data in the new domain violates those assumptions. And did you have any dead ends that are resulted from this kind of phenomena?
This is actually a great question, again, related to medical stuff. So we, in the model, I kind of didn't go into the details of this model from Science Translational Medicine. The paper is available. For instance, one issue that we discovered, which was really putting the performance down, is the calibration of the mammogram machine. That even if you look at the same manufacturer, because you look slightly different versions of the software, the images themselves look different, and it brings the performance down. And in this particular case, we developed, we achieved the invariance by using adversarial techniques. But there are quite a number of other techniques that can be used to deal with it. The main problem is actually to be very explicit in the model, to be aware, and to provide this kind of algorithmic buffers to make sure that you can actually generalize. So in the case of mammograms, because we created all these built-in parts to the algorithm that ensure invariance, it worked well across different population, across different machines. But I am still not 100% confident that if I go to like some population we've never seen, it's going to do some miracles. So that's why we are doing these very big trials to verify that this is indeed the case. And it's not clear to me what is the minimal test you need to do. But the good thing is that there are lots and lots of machinery from the core kind of machine learning part that we can utilize and further develop in medical application space.
Yeah, that's great. And Kevin Layton Brown also asked a question about the generalization to new chemical spaces, because you described a need to generalize to very different tested data rather than using IID samples. And is this practice of the drug discovery community a good idea? Would your methods work better if this constraint were relaxed?
So it's actually a standard practice. And I remember, like, I was not aware of the practice. And I remember in my very early day, MIT is located next to Amgen, which is a pharmaceutical company. And they were members of the consortium. And at some point, we brought our chem probes that we've developed, and we don't, whatever you are using with your handcrafted, fingerprints sucks. Just take our new shiny neural model, and it's gonna do wonders. And we gave them the model, and then it was really surprising. They were nice. They said that it works, as well as their own. It was actually below a model built on handcrafted attributes. And this was the first, I was like totally in shock. And then we realized that the way they do this training and testing, the standard thing for the industry is to do this separation. And you have to bake into the algorithm the ability to generalize, otherwise you overfit to what you see in your training data. And since then, we obviously improved the model, and we learned two things. One is to generalize and to bake it into our designs. And the second one was personal lesson to be humble next time when you talk to areas that you don't know much about. But this is a standard, fairly standard assumption.
Yeah, thanks for the explanation. And another question from Andrew Silva. So in line with your story of getting into the field, how can we get into the space without having medical expertise? It often feels like it's impossible to even properly understand or define problems without having a medical background.
So first I want to say I'm a computer scientist by training. I don't have any medical background at all. But, and you don't need to get breast cancer to get into this field. The lucky part is that there are so many, like it depends what do you want impact. There are problems where you can immediately come in and do impact. There are problems where you need to come up with a new algorithms. So I would say, even though there are not many resources currently available publicly, you already have enough that, for instance, for imaging, there is a lung cancer data set that I mentioned in my set. There are some other data sets. You can just come in and start solving the problem. And you can see that for many of these data sets, our performance is still not sufficiently strong. So you can get into the field. There is really no need to have medical education. And also another good thing, talking about our superpowers, is there are in almost any hospital, if you're a computer scientist, they will come and want to collaborate with you. I cannot promise that every collaborator will be the great one, but if you try a few, you would find the one that you can work with.
Cool, yeah, thanks. And I think given your very inspiring talk, there will be more people wants to get into this field, and these tips are really, really helpful. And there's a question from Yang Qiang asking, how do you handle cross-organizational collaborations, and how do you address the data privacy concerns and fair share of benefits?
Another great question. That's why I highlighted the talk, the paper from Sanjeev Arora that would provide us an algorithmic solution to social problem. So there are areas where it's easier and where it's harder. So for instance, in chemistry, there are already a lot of data sets, maybe not the most developed proprietary data sets, but there are enough data sets you can just come in, they're publicly available like Campbell, and you can start making contribution. It was really amazing to see that, not immediately, but within a few months, there are plenty of data on COVID that you can just go, I'm talking about in chemistry and discovery space, that you can just go in and do research. Now, if you are looking at the space like of mammograms, then it becomes funnier, because despite the fact that we're working with so many places, we actually don't have data in-house from many places. When Adam did the experiments in Karolinska, he had to fly to Sweden, and then he flew to Taipei to do experiments on the Taiwanese data just before they closed for COVID. But sometimes we need to do this kind of things. Now we have a kind of a mechanism where we have the code, we tell to collaborator that's what you need to establish locally to do the training. We kind of created some machinery, but what I cannot answer is what will happen if we take and train the data that come from multiple places. We never actually seen the data that comes from multiple places at once. This is, federated learning and other options to look into it. But I hope I really hope that as AI continue to make strides in this area, there will be more advancement by National Institute of Health and other equivalents in other countries to create and release data sources that come from multiple institutions.
Yeah, and thank you. And another question is about this pre-training or self-supervised learning, because it seems quite common to have this data set with very limited labels or with a huge class imbalance. And you mentioned that it seems like there's not a lot of success stories so far with pre-training. Would you like to share more of your thoughts on this? Like, what are your kind of hypotheses on why it doesn't work, or what do you think might be the promising directions?
So first of all, if you guys, somebody may not believe me, there is a paper from Stanford that was published. You can just, download the code and try it on different data sets. And, in some data sets, it gives you 2% improvement. In other one, it decreases the improvement. So it's, again, when we tried on our wide range of data sets, we saw overall, it didn't really yield the increase. Now, there are several. The way it was applied and people that apply is pretty much to replicate the way we are doing masking in natural language processing. When you remove some part and you try to predict it back or predict the surrounding of certain subpart. So this is my piece of speculation. I don't have anything to prove that this is the case. But one reason I think has to do with how do we get unsupervised data. So unsupervised data is typically taken from something which is called zinc. This is a database, I don't know, of like hundreds of millions of molecules, which are synthetically generated. So it's almost as if we would, have, it's not a random generator because there is some grammar of the molecules, they're well-formed molecules. But it's not what we have in natural language when we are looking at this humongous data that we collect from training, from pre-training our models. They actually have semantic relation. The frequencies of words are reflective of semantics, of real semantics. I'm not sure that this synthetic data is really properly captures, the useful distributions about this data. This is one explanation. Another thing is that maybe the encoding that we are doing with our favorite GNNs is not the right way to create the encoding. Maybe there is a better way to do it and to think about conformance and to move away from the architecture. And I should say that GNN was in the game, in the molecular space for 5 years. It's time to move on. It's not perfect. And so maybe if we make this big leap and change to some other representation, we would be able also to solve the problem of pre-training.
Oh yeah, thank you so much for sharing your thoughts. And I saw a lot of comments in the chat that this is the best talk that they have attended ever. And I also want to share my thought that this is indeed one of the best talks that I have ever attended. And I think you have had a lot of thoughts shared. And there's one more question now from Taeyeon Choi. Like, do you think the current AI and CS communities need to more welcome such multidisciplinary works? Because it seems like sometimes we tend to focus on the novel methods for familiar problems than expanding applications of existing methods to new problems.
So thank you. This is exactly the great question. And the reason before I got personally sick, I never got into the area related to healthcare because I thought, what are these people doing? Just taking a classifier and applying it to the data. There are many people who can do it. So I really hope that in this talk, if there is one message to deliver, that there is a lot of place for method development. It's not about applying. There were the question of pre-training, the question how to model 3D conformers, the question of generalization, the question of even basic representation. Those are hard computational and algorithmic questions for which we don't know the answer. If we just gonna continue, the first generation of models, let's just take what we have and try to reuse and recycle. But if we really want to solve this problem, we cannot do it, and I mentioned a few papers. You're welcome to go and look at the numbers. Every single time when we came up with a new algorithm, with a new approach, with a more sophisticated representation, which is fitted for the task, you do better. And this is my main message, that if we want to really make an advancement, we have to think about these problems as an algorithmic problems. And I would say, I'm with one leg in NLP and another leg in, computational chemistry. And if I'm thinking about, if I'm to compare the two fields, and of course they're very different fields and you cannot directly compare, but what I feel, if I may say, that there are already a lot of kind of standard architectures and algorithms in NLP that kind of are stabilized. Of course, you can improve them, but you're already in some areas in a pretty good space, not to say there's no challenges. However, if you look at what's going on in computational chemistry, from one year to another, you can make an improvement of 10, 15 points absolute in performance. Where did you see it last time in NLP? So there is still a lot of way to bring the field to algorithmically to a point that is satisfactory for us. And despite the fact that, we are very much trying to get there, I don't think that we are quite there yet.
Yeah, indeed. There's a lot of opportunities for these underexplored domains. And also, I want to echo what Kevin commented in the chat, that this is exactly why we have this AI for social impact track at Triple AI. We want to encourage this kind of work with having a multidisciplinary nature, and is kind of exploring the application space of AI instead of kind of focusing on the simply focusing on the methods part. This track features work that talks about using AI and advancing AI for various kind of challenges in different domains. Yeah, so with that, I think we have our time is up. So I want to just thank you again on behalf of all the audience, because this is really, really amazing, inspiring, and touchy talk. Thank you for coming and giving this talk.
Faye, thank you very, very much for sticking with me. And I know that for many of us, it's the deadline night. So I appreciate the double for those of you who listen to me live. And I hope that you all find, an area of AI which will make a contribution to the humanities. Thank you. Good night.